Alright, so we want to talk today a little bit about mitosis, as well as some applications of mitosis. So this is a part of the cell cycle, and specifically this is, when we talk about mitosis, is the main way that humans and most other eukaryotic organisms, in fact, will have cell division occur. So let's take a look here. The cell cycle itself is just a sequence of stages that a cell passes through from one division to the next. Okay, it's going to involve duplication of all the organelles. So think back to Science 10, you learned all about all kinds of organelles. They all get duplicated. What we're going to concern ourselves with mostly in Bio 30 is the duplication of the chromosomes. All right, so a couple basic backgroundy kind of things. Chromosomes themselves are segments of DNA, essentially. DNA is divided up into many chromosomes. The exact number is going to depend on the species. Humans, for example, would be 46 chromosomes. Mice, if I recall correctly, is 40. Different species, different amounts. Fruit flies is 4. Chromosomes themselves, the DNA is usually the part we talk about, but they consist both of proteins and DNA, and that mixture of the two is called chromatin. All right. So the chromosomes themselves can exist in either a condensed or an uncondensed form. The condensed form is visible, and we're going to see them under a light microscope. The uncondensed form is not. And they also can be duplicated or unduplicated. The duplicated chromosomes are attached by a centromere. So that's that middle part. It holds the two chromatids together. And the two pieces themselves are called sister chromatids. But the whole thing, even if it's duplicated, it looks like this weird looking H thing, maybe like a thyroid gland or something like that, at least this one. What's going on here? That it's two separate things. It is one chromosome, because these are duplicates. In terms of the information, we would consider this one chromosome. You'll notice here in blue, genes are not marked out that nicely. In real life, in terms of like being able to see them visibly, they're not like, oh, this is a blue genes, I guess, but that's a terrible joke. Anyways, this gene is a segment of DNA that'll code for a specific trait. Right. Moving on. Maybe. There we go. So I want to talk a little bit about chromosome structure. Now this is obviously a computer image, but it is showing those two duplicated chromosomes and then this fanciful version of the spiral structure of DNA in the middle of it. Isn't quite like that, but gives you the idea anyways. All right. So when we talk about the cell cycle, most of a cell's life is spent in a phase, part of the cell cycle called interphase. And it is divided into 3 parts. There's G1, S, and G2. G1, the G is for growth. We have rapid cell growth. The chromosomes themselves are unduplicated. All right, so G1, there's 1 copy of each chromosome. Easy to remember that one. Some of these, there's not so much, but these ones, yes. Or this one, I should say, yes. This then takes us on to a computer that wants to freeze. There we go. To S. And this is the synthesis of chromosomes, or the synthesis phase. This is the process and the time when those chromosomes get duplicated. And then we have more rapid growth, and this is preparation for cell division. And when we get to the G2 phase, of course, there are 2 copies of each chromosome, right? These sister chromatids that we're talking about. All right, so mitosis itself then. So there's interphase, and then there's mitosis. And the first part of it we consider is prophase. Prophase we can look at it. It is not a snapshot, and it's just sitting around in early prophase, early, and suddenly, boom, it moves on to the next one. Right? So these are, we give 4 general descriptions, and in fact with prophase we kind of break it up into early prophase and late prophase. Early prophase is where we've got chromosomes that are beginning to condense. The centrioles move to the poles of the cells, and spindle fibers begin to form. Okay. Now, the centrioles are protein bodies found in the cytoplasm that allow attachment of spindle fibers. And in a way, a centriole is sort of like the anchor point, like this, on a swing set, right? So this chain has to attach somehow to this end. All right, that's going to be at the centriole. All right, and then we've also got kinetochores, which form on the surface of the centromeres, and this is the other end. So think the swing end of this chain. The chain is like the spindle fiber. It's going to guide the chromosomes, and this end here is attaching in the cytoplasm. And then the other end, the swing end, is attached to the chromosome at the kinetochore. The spindle fibers themselves are protein microtubules that attach to the kinetochores, and they're going to guide chromosome movement during cell division. All right, that's early on. So this is all starting to happen. We've got a picture coming up right away. Late prophase, the chromosomes continue to condense. All right, so what's happening here is they are almost like they're coiling up. So we say that they're condensing, and that's because they become easier to see. But the centrioles themselves are going to continue to assemble. Spindle fibers are going to attach to the kinetochores, and then this is important, the nuclear membrane begins to dissolve. And this is how we know we have reached late prophase, is that that nuclear membrane is dissolving. So, moving along. Potentially. Very good. Some diagrams here. And we'll look at the 2 of them here to see how they're different. So early prophase, that's kind of on the top here. You'll notice nucleus is still intact. All right. Up here we've got them, our centrioles. They are starting to assemble. They're going to arrange themselves kind of on the pole. We call it the poles of the cells, opposite sides essentially of the cell. Note though that the nucleus is still, the nuclear membrane I should say, is still intact, but we do have these chromosomes. And now if we were to look at this particular cell, it has 3 chromosomes. Not typically common to have an odd number of chromosomes, but for this particular example, 3 is kind of nice because it fills up the space without being onerous to look at. Now, when we get from early prophase to late prophase, what do we see? Well, we see that this nuclear membrane has broken apart. Okay, so these bits here have now broken apart. They're scattered throughout the cell. The microtubules, right, these spindle fibers, have attached at both ends. So early prophase, they're attached more or less at the centriole end. By late prophase, they are attached not only at the centriole, but also at the centromere, so in the middle of our chromosomes. All right, which then takes us on to, well, what happens next? And our next step along this little journey is going to be metaphase. During metaphase, the chromosomes are lining up on the equatorial plate. Oh, I should also just point out, going back here 1 minute, I believe your textbook or some of the diagrams you already have refers to prophase as early prophase, and then this late prophase they're going to call prometaphase. And so different terminology meaning the same thing. as in early metaphase instead of late prophase. So during metaphase, the chromosomes line up on the equatorial plate. The nuclear membrane dissolves completely. At this point, the chromosomes are actually kind of entangled. In our diagram here, we can count them pretty easily. In a real cell, not quite so much. They're not easily counted at this point. All right, and so we see, once again, this time instead of as in late prophase, where they're attached to the spindle fibers but not lined up nicely, here we've got them not only attached to the spindle fibers but lined up nicely. All right, this is metaphase. They're lined up in the middle. It's very easy to see metaphase. And we've got anaphase. And here we've got the centromeres are dividing. And these sister chromatids, which we now have to call individual chromosomes, are going to move to opposite poles of the cell. Occasionally, segments of chromatids might actually break apart and then reattach during anaphase. And what do we see going on here? Well, we see the centromeres have divided. Okay, and so now the two halves of each sister chromatid, which again we now have to call separate chromosomes, are moving to either side of the cell. You'll notice the cell itself is elongating here. All right, and so this is leading up obviously to eventually we're going to have division happen here. But this is not going to happen during anaphase. But that you can see we're leading up towards that. That's what's going to happen. Right. So let's move on. You can see here when that dip becomes more pronounced, we're going to call this a cleavage furrow. Because this is where the two cells are going to split apart. The one cell will split apart and become two. Right. Telophase. At this point, the chromosomes actually reach the opposite poles, and then they begin to lengthen, or if you'd rather than lengthen, they begin to uncondense. The spindle fibers themselves are going to dissolve. The nuclear membrane forms around those uncondensing masses of chromatin. So all of those chromosomes are uncondensing. They're becoming long and spindly, hard to see through a microscope by the end of telophase. And a new nucleus forms around each of them. All right, cleavage furrow has become more pronounced at this point. And the spindle fibers are still there, but they are starting to dissolve. They have let go, generally, by this time, of the centromeres. And they are eventually going to be dealt with by lysosomes in the cell, and then the proteins, actually the amino acids, which would be the building blocks of those proteins, are going to be able to be reused for other cell functions. And then the last part here is not normally what we call part of the chromosome part of this thing here, but this is actually when the cell itself divides. Okay, and this is called cytokinesis. It's the division of the cytoplasm. In animals, this is begin with the furrows developing, like we've been showing, which will actually pinch the cell into two parts. Plants, generally the cells don't form a cleavage furrow. What rather happens is a cell plate grows in between the two halves of the cell, which will then develop into a new wall, and it seals the two parts of the cell off from one another. Right. So you can see here we've got a cleavage furrow formed. Over here, on the other hand, we've got a cell plate forming. And you can see some of these cells. Here we've got this one is in telophase. So we've got up at the top, there's a nucleus. There's another nucleus. These are both newly formed nuclei. And the cell is in telophase, and it's much fainter. You can see it here. It is much fainter, that cell wall is, than the other ones that surround it. And some of that comes down to the fact that we know what we're looking at here. One of the reasons we can tell, though, also that this cell is newly made, the nuclei are not as big, nor are they as indistinct as in the other cells. All right. A couple applications here. One of the big ones that we're going to talk about is cloning. And cloning is where we end up forming identical offspring from a single cell or tissue in the parent. And so the clone then will have identical or nearly identical DNA to parent cells. Some clones will have accidental DNA changes, or if we started with a potentially mutant version of a parent cell, of course, then the new clone is going to be identical to that one, which might not be the same as the rest of the parent. The other thing that obviously can happen is some mutation might happen during that cloning process in the new cell division. All right, so cloning itself is a form of asexual reproduction. It does occur naturally in Hydra type of organisms, some plants. And I guess we can say that identical twins, or monozygous, starting from one zygote, those type of twins are clones of each other. Now, this is naturally happening. It's not like we've got somebody trying to make that happen. In plants, a little bit of history here. A guy named Frederick Stewart cloned a carrot from a single carrot cell in 1958. His method works for some types of plants, but not others. Okay, in terms of you knowing it, this is giving you some background. I'm not expecting you to know this guy's name and the year, nor the reason why it works for these. And no, so for orchids, carrots, ferns, tobacco, petunias, lettuce, but not grass and legumes. Potentially a research project for the end of the year. And again, it produces offspring with identical characteristics of the parents. All right, what about animals? Well, Robert Briggs and Thomas King cloned grass frogs using the process of nuclear transplantation. This nuclear here is referring to the nucleus of the cell. This is not some kind of radioactive thing. We're not creating that type of a clone that is somehow now going to become a superhero or something like that. They used a micropipette or a small glass tube to remove the nucleus of unfertilized egg cells. All right. They implanted the nucleus from a frog embryo that was in the blastula development stage into the enucleated egg cell. So enucleated meaning an egg cell that's had its nucleus removed. All right. So if we want to think about it this way, actually, come on. There we go. Occasionally, these would grow into adult frogs, which were clones of the donors of the nuclear material. So what we would have is we have a frog egg cell, and we take the nucleus out. All right, so we're going to assume this is the nucleus. We are removing that nucleus, which gives us an enucleated egg cell, right? An egg cell that has no nucleus. Now what we do is we take, here's a frog zygote, and it's actually developed into the blastula stage. So we're going to take the, and I'm not going to try to draw a whole blastula here at this point, but we're going to take the nucleus from one of those blastula cells. Okay, so this was egg cell from frog number 1. Here's the egg cell again from frog 1. Here is the result of fertilization from frogs number 2 and 3. All right, and so now we've got the egg cell here, or the nucleus here. We're going to take this nucleus out and insert it into the nucleus or into the egg cell from frog number 1. Right, so this one no longer has an egg cell. Right, this is the this was that blastula stage from the, we're going to call this frog number 2 and 3 blastula. We take the nucleus there, put it into this enucleated egg cell. Occasionally, Briggs and King were able to get this cell to now grow into an adult frog, which I'm not going to attempt to draw on my computer screen. And this adult frog would be a clone of this nucleus here from frogs 2 and 3. All right. However, if they waited to the gastrula stage, mitosis in the new egg cell did not occur for a long period of time, if at all. Now, this suggests that as cell specialization began, right, so by the time we get to gastrula stage, we're looking at the 3 germ layers, right, the mesoderm, the ectoderm, and the endoderm. And by that time, cell specialization has begun, and so they're less able to undergo mitosis. All right. Now, adult mammal cells are very difficult to use in cloning. Until recently, anything beyond the 8-cell stage was very difficult to take that nucleus, put it into an egg cell, and make it start doing cell division. So even like, 16-cell, which we'd still consider morula stage in mammals, very difficult to make that happen. There are lots of chemically monkeying around with it that we have to play and do. Dolly the sheep, exception to the rule here, was cloned from the nucleus of an udder cell from an adult, okay, this is the breed Finn Dorset sheep. All right, nucleus from this udder cell was placed into an enucleated egg cell from a Pole Dorset sheep, developed in vitro to the early embryo stage and implanted into the uterus of a 3rd sheep. So again, if we were to kind of diagram this out, we take a, here's my udder cell. We're going to take the nucleus from this one eventually. Okay, so this is Finn Dorset sheep. We're going to call this sheep number 1. And then over here, we've got, so this is number 1. Over here, we take an egg cell. This is from Pole Dorset sheep. This is sheep number 2. And this is the naturally occurring nucleus. We're going to get rid of that nucleus, binucleus, which forms our enucleated egg cell. Okay, and again, this is the egg cell from the Pole Dorset sheep. This is sheep number 2. All right, we're going to take the nucleus from sheep number 1, put it into sheep number 2's enucleated egg cell. There we go. And we allow this to develop a bit, and we are going to then implant this in the prepared uterus of sheep number 3. Okay, looks more like I'm not sure what it is, but it's going to, so we take this whole thing, it goes into the uterus of sheep number 3, and lo and behold, we get a viable lamb out of this deal. All right. The genetic information of Dolly, okay, so that's the lamb that was born from sheep number 3, was shown to be identical to the genetic information in sheep number 1. Now, problem with cloning, at least with Dolly. Dolly was the first success in 277 attempts. So this was not a high percentage probability thing to happen. This took a lot of work. It did not work out well for the first 276. Potential benefits. By cloning, we have an increased ability to interpret drug effectiveness in test animals. Right? The drug would affect all identical animals the same way. We could also clone our strongest or best livestock, which would increase yields and decrease losses. A picture of Dolly. Couple things here about cloning. One word we should talk about is this idea of pluripotent, because a pluripotent cell can turn into many different types of cell. Normally, right, if you were to take and you cut your skin or something like that, or scrape your skin, new skin cells will regrow. But you're not going to randomly have taste buds grow on the side of your arm, or retinal cells. It's going to grow into one type of cell because the cells in your arm are not pluripotent. So normally, some of the DNA in differentiated cells is inactivated, and that means the cell can only turn into one type of cell, which means these cells are not pluripotent. Research has shown that the reason that cloning in Dolly and other mammals can be successful is due to factors or chemicals which we find in the oocyte. In 2006, scientists were able to artificially change a differentiated cell into a pluripotent cell using a mixture of 4 chemical factors. And so they actually were able to create a clone without any nuclear transfer. More recent research has shown that some types of human skin cells, by messing around with these chemical factors, can actually be changed to become nerve cells, which, given that central nervous system damage normally is hard to fix, this shows potential promise for using skin cells to create nerve cells. Right, we're going to stop there. We'll have some work to do.